Hello again, everyone. Welcome to my class on biochemistry, where we try to simplify biochemistry in its simplest unit. So, today I'll be talking about biochemistry. Carbohydrates and its significance biologically. And I expect that at the end of this class, that we will be able to define carbohydrates, differentiates between the different types of carbohydrate that we have, and also look at some of the bonds that makes the monomers come together to form the different forms of carbohydrate that we have, and analyze the biological significance of some of those monomeric and polymeric units. So forms of carbohydrates, before I go to that, let me just quickly touch on what carbohydrate is. Carbohydrate is a carbon-based molecule that is rich in hydroxyl group. Let me just put it that way. So we have C and an OH. Whenever you have that group, then you know that this is a carbohydrate. With empirical formula CH2O, then the multiples of the N. And carbohydrate can be simple and it can also be complex, and that is what the N defines. When N is at its very low numbers, then we're talking about simple carbohydrate. But when it becomes complex, there are different bonds that come into play, and then we have the multiples, which is the N, to be higher. Carbohydrate primarily function as energy storage and also for structural support, and as well as cell recognition. Now, carbohydrates can dazzle in different isomeric form, and the simplest carbohydrate is glyceraldehyde, which has just one chiral center. The more the chiral center, the more complex the carbohydrate is, and the chiral center is defined by the isomer, the epimer, D, and the L form. The D means that it is dextrorotatory, which means it is rotating to the right, and the hydroxyl group is on the third, especially the third carbon atom. Or most times the chiral center is on the right, while for the level rotatory is on the left. So the position of the hydroxyl group for the reference carbon usually determine if it is a D isomer or an L isomer. Now let's talk about different types of carbohydrates, starting with monosaccharide. Monosaccharide is the simplest carbohydrate that can't be broken down any further. Examples are glucose, mannose, fructose, galactose, and it can exist in linear form as well as in cyclic form, but the most common in nature is the cyclic form. That's the Haworth projection when it is in water. Now, examples of monosaccharides are, as I mentioned earlier, ribose. We also have the oxyribose, which is the carbon 2 having no oxygen, and this is the common one we have in a nucleic acid structure. We have glucose, we have mannose, and others. And this is their fission structure. Like I mentioned earlier, the Haworth structure is more common. This is how the Haworth structure is formed from the fission structure. So look at this fructose now becoming cyclic by losing some amount of hydroxyl ion, and also forming the alpha D fructofuranose. This is the cyclic form of glucose. Because it has become cyclic, we call it fructofuranose. It has the furanose shape at this point, the furanose shape. I'll go to the next slide, which talks about the different types of carbohydrate, now focusing on disaccharides. Saccharides contains 2 units of monosaccharides coming together, and there is a linkage between this unit. This linkage is generally called the O-glycosidic linkage. Examples of disaccharides that we're familiar with is lactose, which we have in our milk, maltose, which we have in most of our starchy foods, sucrose from our beets or cane sugar, and also cellulose. But cellulose is more of a complex carbohydrate, but sometimes it starts from the 2 units that makes it up. So going to the next one, we have examples of disaccharides and how disaccharides are formed. Now, I will take a pause to explain something like this. We also have oligosaccharides. That's when we have 3 monomers coming together. But In this case, I'll just pick monosaccharide, disaccharide, and also polysaccharide. Because many sugar has hydroxyl group, they can interact with one another. So when you have for example, let me look at this maltose. Maltose is made up of two glucose units, but they are linked by alpha 1 4 glycosidic linkage. So this alpha is defined by, so the first carbon is usually referred to as the alpha carbon that carries the hydroxyl group, the aldehyde group. And now, so we have this carbon serving as the alpha 1, 2, 3, 4, 5, and then 6. That is how this structure is labeled. Then the second glucose has been flipped because we want to form a bond between the first and the fourth carbon. So it has been flipped to have it as alpha 4, and we have this 1, 2, 3, 4. So when these two come together, we have alpha 4 glycosidic linkage. This also applies to fructose, where we have dehydration will definitely occur for this bond to be formed. There is a process called dehydration where a molecule of water is released, and then these two come together. For fructose, we have alpha 1 beta 4 glycosidic linkage, yes. So in this case, we have alpha 1, but in the case of fructose, because we have fructose to have a furanose structure, not the pyranose structure. The furanose structure of fructose, the keto group is on the second carbon atom. If you look at the Fischer structure that I displayed at the first time, there is a difference between glucose and fructose, even though they are both 6 carbon units. But the second carbon atom carries the keto group, while glucose carried an aldehyde group in the first carbon atom, and this glucose is an aldehyde, and fructose is a keto, contains a keto group. And I think you can do more and just read up on some of these things based off some very simple texts like short course in biochemistry by Stryer, Huber Stryer. So the next one, we'll talk about some of these very common examples, and then I take more reference to what we are familiar with. For sucrose, it's made up of two units, which is glucose and fructose units. As I mentioned, if you look at it, the alpha 1 and the beta 2 linkage. This in this case now, because this is the carbon that is carrying the this is the carbon that is, this is the first carbon that is carrying this CH2OH, and then we have the second carbon which is carrying the keto group. Now there is the formation of the glycosidic linkage between the first and the second carbon, but in this case it's beta, not alpha. So that is why for sucrose we have alpha 1 beta 2 glycosidic linkage, and for lactose we have beta 1 4 glycosidic linkage. In this case, the first one is galactose. Lactose contains galactose and glucose, and the linkage is between. If you look at the structure of galactose, the OH is on the fourth carbon atom, but in glucose we have the OH where the OH isn't. So this linkage between for this linkage to work, there is need for a switch in the position, and that is why we have it as alpha 1 for glycosidic linkage, and this can only be acted upon in human by lactase. When we take milk, we have this lactase in our milk broken down by lactose in our milk broken down by lactase, and in bacteria it is broken down by alpha galactosidase. While for sucrose we have sucrase breaking it down, and for maltose we have maltase cleaving it during the process of digestion. Okay, so the third one that we'll look at is polysaccharide. They are long chains of monosaccharides, and they can be linear or branched. Examples include the starch that we have in plants, which contains alpha amylose and amylopectin, and glycogen in animals, and also we have cellulose in plants. These are just common examples. Now, these two chains of glucose molecules that are joined together by alpha 1 4 glycosidic linkage, as well as alpha 1,6 glycosidic bond. The alpha 1,6 glycosidic bond is what is responsible for the branching in glycogen. This is the structure of glycogen. This is the unit of glycogen that was at the point of branching. And alpha 1,4 is responsible for the linear part, then it branches at every 10 glucose units. So this branching is taken up by the alpha 1,6 glycosidic bond. And as I mentioned earlier, this is what is responsible for its ability to store, to perform the function of storage. And one of the things that I want you to note is that glycosidic bond determines the function or the structure of polysaccharide. So if it is very linear, then we have it doing more of structural function, while if it is more bent or we have more branches, then it's more suitable for storage. And this is what we have, for example, in cellulose we have more linear bonding, while we have starch and glycogen being somewhat branched, and so that is why they function more as storage polysaccharide. Now let's look into the difference between glycogen and starch. Glycogen and starch are both glucose polymer, but glycogen is more branched and it's found in animal, while starch is less branched. I think starch is about for every 30 units. I'm not quite sure now. But glycogen is more branched and it is found in animal, so much tightly packed. Amylose and amylopectin in glycogen is what is responsible for its branching, because amylopectin has about the 1,6 linkage at every 30 unit that pass of alpha 1,4 glycosidic linkage. So moving on to the next slide, I want to quickly look at some complex polysaccharides aside cellulose, starch, and glycogen that we talked about. We have glucuronic acid, N-acetylglucosamine, and these are some glucuronic acid, N-acetylgalactosamine, sulfated glucosamine, and also sulfated hydrochloric acid. These are some very complex polysaccharides that function in some very salient, but and I think you'll still get to know about some of these things. Some of them are in as act as lubricants in the mucous system, and also in some of them even in the blood system. We have all our immune system, the ABO blood group system, and the like. And also we have some non-starch polysaccharides like chitin, which is responsible for the exoskeleton that we have in some of our invertebrates. Cellulose, structurally its function in plants, and also pectin, which is also something that is that we find in most of our fruits, is a polymer of galacturonic acid that is linked with some galactose and arabinose. Yes, and arabinose that are branched together, and sometimes I think it's also partially methylated. If you also look into the structure, it has this methyl group. This is what is responsible for the pectin structure. So this is inulin, and inulin it also has this already branched shape, which makes it to also function in some structural and storage function as well. Now there's something that I want to quickly look at, which is some glycoconjugates. We have carbohydrates covalently linked to other biomolecules like protein and lipids. So looking at protein, when protein links to carbohydrates is called glycoproteins. Glycoprotein include the general protein the general glycoprotein, and this means that it is dominated by protein. We have more of protein than the carbohydrates. But another glycoprotein is protoglycan. Protoglycan contains more of carbohydrates than protein, and so I think the third one that is also very common is mucoprotein, which also has more of carbohydrate than protein. And an example is the mucin that we have in our mucus, which forms the major lubricant function, or contributes to the major lubricant function in our mucus. Now the second glycoprotein, glycoconjugate I want to look at is glycolipids, which is quite ubiquitous in most of our cell membranes, and it plays a role in cell-to-cell interaction. Simply, the simple form of glycolipid is cerebroside. So some of the examples of glycoproteins that I mentioned is glucosamine, galactosamine, chondroitin 6 sulfates, and the likes. So these are some of the places where it abounds and that it functions in hormone regulation. An example is erythropoietin. Now let me quickly look at the biological significance of carbohydrates as a whole. Carbohydrate has diverse roles, like energy storage, as we see in glucose and glycogen, structural components, as we say in cellulose, and also in cell recognition, like we say in glycoproteins, forming major components of membranes, and also a major component of nucleic acids, like ribose or deoxyribose in DNA and ribose in RNA. Now, I would just quickly touch on the physiological significance of some hexoses. We have glucose, we have fructose, we have galactose and mannose. These have their own biological significance as well, because they are readily metabolized. For example, when we have some disaccharides, they are easily cleaved to give the monosaccharides that then enter into different pathways to generate energy. And some of these hexoses also form some complex interaction with other biological molecules like proteins to help in the maintenance of biological membranes and biological fluids. Pentoses also has their own significance in the sense that it contributes to form most of the components of glycoproteins too, and sometimes we use them as to detect some defects in any pathways, in some pathways. For example, for G ribose, we have it in ATP, in NADPH, in flavin coenzymes in the citric acid cycle, as well as in the electron transport chain. So finally, I'll just give you an overview on various sugar that are formed in human tissues, and these are just some of the different layers. Just to wrap up, we have 3, 4, 5, 6, 7, 8, 9 carbon. So when you have it as 3 is triose, 4 tetrose, 5 pentose, 6 hexose, 7 heptose, 8 octose, 9 nonos, and these are different examples of carbohydrate that monosaccharides that has this number of carbon units, starting from glyceraldehyde. So examples of various forms that some of are familiar with, you can just check on N-acetyl glucosamine. Is a very important polysaccharide that is found in human tissue. We have glucose 6-phosphate, also very important. When we start talking about some of these pathways, you get to know more about this. And so summarily, monosaccharides are the simplest carbohydrates, and you have to also know that for them to form complex carbohydrate, there has to be a linkage, and most of these linkage are all glycosidic bonds for every of the structural components of living tissue, cellulose. So these are some of the references that I used, and so I hope this video has been able to enlighten you about the basics of carbohydrates and some of the structures now. So please leave a like on our video if you find it helpful, and also subscribe to this channel so we can have more of this video come to you the moment we have it published.